Okay, Assalamualaikum warahmatullahi wabarakatuh. We are from Group 5. We present about Basal stem root diseases and the pathogen is Ganoderma boninens. Okay, for the taxonomy, the domain is eukaryote, the kingdom is fungi, the phylum is Basidiomycota, the order is Foliformes, family is Ganodermataceae, genus is Ganoderma, and the species is Ganoderma boninens. Okay, for the first part is pathogen life cycle. There are 10 stages that I will explain. The first part is the basilospores which germinate into the monokaryotic mycelium and then plasmogamy will occur and produce the dikaryotic hyphae. And the second stages, you can see there are clamp connection to maintain the new state of the mycelium. Okay, for the third to fourth stages, you can see the basidial cup is formed. In the fifth stages, the layer of hymenium is fully formed. Okay, for the sixth stages, the basidium is formed, and the seventh stages, charigamy occur in the basidium, and after the meiosis, you can produce four haploid nuclei. And the ninth stages, you can see four protrusion, which we call also by the spore formation. And the 10 stages, you can also see there are four by the spore are formed. Okay, that's all from me. I will pass to my friend to explain about the disease life cycle.
Okay, hello, Assalamualaikum. My name is Muhammad Hadi bin Zulkifli. My matrix number is 200785. So today I will presenting about the disease cycle. So the cycle of the Basal stem root. Okay, so I will simplify the Basal disease cycle of the Basal stem root with the by the progression and the symptoms of the disease. Okay, so the disease progression first is sort of reticulum for Basal infected root to healthy root contact, and then the infected debris in the plantation and basilospore. And we can see in this first progression is we can see no symptoms in here. And the second is root colonization by Ganoderma boninens. And the symptom we can see is the chlorosis of leaf. Okay, so next slide. Okay, and then we can see the basal stem root progression in base system. And the symptom we can see is fully elongated unopened leaf. Then for the progression, complete colonization by Ganoderma boninens, and after progression in base system, complete colonization by Ganoderma boninens in base with restricted water uptake. And the symptom we can see is lower leaf collapse hanging downwards, giving skirt-like appearance. Younger leaf group turn yellow and die back from the tip. Okay, next. And the the next progression is base of the stem turns black, degradation within the stem, and we can see the symptom is the appearance of the basidial cup, and lastly the crown of the palm fall off and stem collapse, and we can see the oil palm dead.
Okay, so now we will move on to the next part about the first symptom. So these pictures show the early symptom of the Basal stem root disease in young oil palm, where the lower leaf become yellow or develop yellow and green irregular spot called as mottle, and then the leaf bend downwards and begin to rot prematurely, which later the growth becomes slow, palm become pale, and the spare leaf remain unopened. So next, photo on the left side show the disease infect the mature oil palm tree. As we can see here, the lower leaf collapse and hanging downward vertically from the point of attachment to the trunk, which make a skirt-like appearance. While on the other side show the internal symptom of the disease, where we can see it appears as a light brown dry rot lesions. And then next, we can see the new ganoderma start to form at the base of the oil palm as shown by the arrow, which the size can go up until 6 cm or above. Then we can see the color is brown on the top and white on the below. And then later, the basal rot in oil palm can cause the trunk to collapse, and the oil basal cup will form on the roots.
Alright, now is the diagnostic test available for Ganoderma boninens. To diagnose Ganoderma disease, we can use polyclonal antibodies against the crude mycelial proteins of G. boninens by indirect enzyme link immunosorbent assay ELISA. So here is the overview in doing the assay. Firstly, collection of Ganoderma pure culture to produce antigen. Second, growth in liquid medium. Third, protein extraction. Fourth, immunization and polyclonal antibodies production in rabbits. Fifth, sera collection. And finally, ELISA. So, how does enzyme link immunosorbent assay polyclonal antibody works in diagnosing Ganoderma disease? First, Each column of the ELISA microchip plate is coated with the antigens. Second, the plates are rinsed with phosphate buffer saline with 0.05% PBST 3 times and then blocked with 5% skim milk at 37 Celsius. Third, then zero dilution of the primary antibody prepared in a SEPA for PBST are applied down each column in fifth fold. The plates are again washed with PBST and the secondary antibody applied in 50 microliters. After 1 hour incubation, the plates are put through the final wash cycle with PBST. 6. 50 microliter Horace peroxide, conjugate gold anti-rabies immunoglobulin is added to each well and incubated at 37 Celsius for another 1 hour. 7. Add 50 microliter for each well with enzyme benzethanoic sulfonic acid and follow with addition of 50 microliters of 2 molar H2SO4. 8. Absorbance of the hydrolyzed substrate is measured at 405 nm with an ELISA reader. Sample is considered positive for Ganoderma infection when the platelet graph not fall on the standard curve. So that's it, how ELISA PAD works in diagnose the Ganoderma disease.
So now I will be talking about the management options for Basal stem rot disease for the Ganoderma bonninense. First, we have the physical control. Sanitation is the basic measure to control BSR. This can reduce the dispersion of the pathogen inoculum. Infected palms, thrombosis, and rings that is infected by this pathogen must be removed and destroyed from your condition. Perform surgery mounting of Ganoderma infected palms can extend its lifespan, applying protectant chemicals such as Therum to treat the exposed area from decaying. Then we have the chemical control. Applying fungicides with the soil mounding method. If the infestation is heavy, immunization of the planting materials using benzoic and salicylic acids are proven to decrease the BSR emissions. Use mineral fertilizers such as calcium nitrate and trichoderma species, a biocontrol agent, can suppress the BSR disease. Adequate supply of minerals to the oil palm such as calcium, copper, and salicylic acids can improve the plant resistance towards the disease. Next, we have the organic approach. Using the fungi hypnum species and Chlorotus bijamar can enhance the degradation of food materials for the oil palm. Other option using the lenzites and marasminus, but this one is a risky one because it might become another disease factor for the oil palm. Then we have the white rod hymnomycetes that can be used to increase the rate of degradations. This study shows that this one can help to increase the mass loss of the infected oil palm and eventually reduce the inoculum pressure. Then, moving on to the biological control, biocontrol application using fungus and bacteria on the necessary state can reduce the incidence of BSR. For example, we have the Hendersonia thorolide gano EF1, then inhibiting the BSR disease by using endophytic microorganism. Also utilizing the antagonistic effect of Ganoderma bonninense using the actinomycetes. For example, we have these Nocardiopsis species, Streptomyces. So this is the inhibition rate. Lastly, we have the resistant variety. So resistant breeding has not yet been discovered fully because there's limitation, for example, plant resistant source and effective screening technique for now. So partial resistance has been developed using the cross of Zaire and Cameron from the NPOV's semi-wild germplasm. So the downside of this method, it took a longer period to be established, as backcross breeding process took about 30 to 40 years. Variety average oil palm, compared to commercial varieties, is proven to be resistant toward the Ganoderma bonninense. So how management option can affect the disease progress curve? So in physical control, eradication reduce the production of initial inoculum by destroying or inactivating the sources of initial inoculum. Then in chemical, protection reduce the level of initial infection by means of a toxicant or other barrier to infections. Organic approach, avoidance, reduce the rate of reductions of inoculum, prevent infection, and deters any developments of the pathogen. Biological control, eradication reduce the initial inoculum and inactivating the source of initial inoculum by suppressing the pathogen using biocontrol agent. Lastly, the resistant variety. Resistant is using cultivars that are resistant to initial infections, or planting cultivars that can reduce the rate of inoculum productions, infection, or pathogen development. So that is all from management options. So here is our list of reference. And so that is all from Group 5. Thank you.